Alright guys, today we're going to look at protein synthesis, okay? We saw the idea of DNA and RNA. Those are two different kinds of nucleic acids, right? DNA is two strands, right? RNA is one strand. DNA has adenine, cytosine, thymine, and guanine. RNA has adenine, cytosine, uracil, and guanine. And instead of deoxyribose that DNA has, RNA has ribose sugar as a part of its backbone. Okay? Well, the whole point of RNA is to help us to make proteins. That's kind of the key thing here. Okay? So that's called protein synthesis. And there are two basic sections to protein synthesis. The first section is called transcription. What is transcription? Easily defined. What we're doing is we're taking DNA, a little section of it, and using it to make an RNA sequence, okay? So we're taking the DNA, and again, remember, DNA is a very large, long molecule relative to RNA. We're going to take a section of that DNA and make a small section of RNA from the DNA. That's transcription, okay? And then the second section is called translation. This is where we take the mRNA, we put it on the ribosome, and then it tells us what amino acids to stick together to make a protein. Okay? It's literally the assembly of the amino acids into the protein. Okay? Now, you got to do this one before you can do. You got to do transcription before you can do translation. Okay? So what we'll do real quick is we will look at transcription. What is transcription? Everybody in here has heard of a transcript, haven't you? Okay? A transcript is a written version of what's going on. Like if you move from one school to the next, right, you have a transcript. It's a written piece of paper that tells the new school what classes you took and what grades you ended up with in them, okay? That's a transcript. So we're writing a transcript of the information on the DNA. But we don't need the whole DNA strand, we just need a little section of it, okay? So during transcription in the nucleus, a segment of DNA unwinds and unzips, and the DNA serves as the template for the messenger RNA. Okay? It's going to be, we're going to use this section of the whole DNA strand, and we're going to use it to build a messenger RNA molecule. Okay? Now, we also have to have an enzyme to build the RNA, and it's called RNA polymerase. Remember, with DNA, we had a DNA polymerase that built the opposite strands of DNA. Here we've got an RNA polymerase that's building the opposite strand on the DNA to make RNA. So it is an enzyme in the nucleus. It does a couple things. First off, it separates excuse me, the two DNA strands. Okay? It literally goes like the helicase did. Okay? And then it moves alongside one strand, not both strands of the DNA, but just one of the strands, and it makes an RNA sequence of nucleotides. It joins them together. Okay? So, here's basically what we got. We got the DNA, we got these two stripes here. This is the double helix. The RNA polymerase is running, is running down, okay? It's running down the DNA. And as it goes down, it's building a mRNA piece here. It's adding mRNA nucleotides. Here's a little bit more detailed version. So we've got the DNA here, the purple backbone here, C, G, A, and T, right? We split it, and just on one side, we're building this. And you see the DNA strand is a little bit different looking than the RNA strand. First off, it's only one strand long, not two. It's got a different sugar, that's why this string is not purple, but it's kind of blue. A and T, C and G, G and C, C and G, U and A. Remember, we do not have thymine. You will not see T on this RNA sequence. But on the DNA, you see the T. Okay? And again, the RNA polymerase is building that strand of RNA. Okay? So I have a question for you. Okay? Real simple, real straightforward. What is going to be the complementary RNA strand for the following DNA sequence? Here's my DNA sequence. Looks familiar from the other day, doesn't it? C, G, C, T, A, T, G. Well, what's complementary? Well, we saw with DNA, it would be CG, CATA, G, right? But this is RNA, it's not DNA. So here's that strand again. The opposite side would be this. C, G, C, A, U, A, C. There's no T there, it's always uracil, okay? That's the main difference between RNA and DNA as far as the sequencing. There is no T in RNA, okay? Always remember that. There is no T in RNA, okay? So, that simply put, and I do mean simply put, that's transcription. Now you're going to see, I've got a couple of videos that I'm going to post on the YouTube channel that's going to have not only these notes, but it's going to have some examples, some videos, 3D animation of how transcription occurs. And you need to watch them and make sure that you understand what's going on with the transcription. Now make sure if you have questions to email me, okay? So don't forget to email me, okay? All right.